There were a number of good abstracts across the spectrum of acute myeloid leukemia presented. I think we saw some very important extended data presented from the VIXIOS phase 3 study, looking particularly at patients with the post-myelodysplastic syndrome subtype of acute myeloid leukemia. And those results showed that in that subgroup, which accounted for about 2/3 of patients in the international study, they all stood to benefit from, in a similar way to the wider group, from the use of CPX351. Some other very interesting oral abstracts that were presented included some laboratory rationale for development of CD200 targeted treatment in AML. CD200 is an immune checkpoint which potentially sends an immunosuppressive signal in AML, which stops the patient's own immune system dealing with the disease. So targeting CD200 with antibodies is one of a number of potentially attractive approaches to harness the patient's immune system in fighting the disease. And one of the postdoctoral researchers, Dr. Rastogi from Cardiff, yesterday presented some interesting data with an interesting animal and basic science preclinical model showing that targeting CD200 can harness this endogenous immune activity. So that's something which will hopefully be taken through to clinical trials in the near future. There was also some very interesting data presented on some new epigenetic targets in AML as well. So there's a whole wealth of new targets that are sort of following behind the recently approved new drugs.